sorry. Technology sometimes. So I think we got the results for exam 2, and you guys actually did it very well. I think average is, mean is a little bit higher than the exam 1. So I'm not sure. Do you guys see the scores? No? Oh, okay. Well, TA probably will post it today. All right, so today we're going to talk about the protein metabolism. And Thursday and the next week, Dr. Carrie Ersman is going to give a lecture on the amino acids metabolism, right? So protein synthesis and degradation. So the protein function is a little bit different from the lipids and carbohydrate. Proteins constitutes about 17% of our body mass, right? And functions, the protein, I think it's very important, right? So for example, a protein maintains the body's structure, the collagens, basically is proteins. So, and then also it's very important for facilitate mobilities. For example, in the in the muscle, actin and myosin are all proteins, right? They're very important. And transporter. We know that hemoglobins actually transport oxygen, and a lot of membrane transport protein systems like glucose transporters, amino acids, and transporters, and fatty acid transporters are all proteins, right? So protein is really very important again, and metabolism. A lot of enzymes, involve the different metabolic pathways, are also enzymes, and regulations. The protein involved in very important regulation of metabolic pathways. For example, hormones and growth factors are all proteins, and also immune function, right? Also involving immune function, like immunoglobulins and cytokines, and all the proteins. And protein amino acids can also be used to produce energies, right? So protein, has a lot of very important biological functions. And here's the overview of protein and amino acid metabolism. And in our body, we have a amino acids pool. And amino acids in the body can be from the, be coming from the diet, right? We eat proteins, and diet and protein that can absorb the amino acids into the body. And also our body has a protein stored, right? And then protein can be degraded and also contributed to the amino acid pool in the cells. And amino acid can also be used for synthesis of proteins. So protein turnovers mean protein synthesis and degradation need to be balanced in the body, depend on the physiological state or depend on the needs protein, in the body. And in terms of a metabolism of the amino acids, we know that amino acids contains carbon and also nitrogen, right? So there are actually two aspects of metabolism of the amino acids. One is the carbon metabolism, the other is nitrogen metabolism. So for the carbon metabolism, amino acids can be utilized for energy and during, for example, the very fasting conditions, right? We need to utilize the stored energy. We already talked about carbohydrate like glycogen, and also fat, right? That's a triglyceride. Fatty acids can be mobilized and for energy, but in addition to glycogen and triglyceride, and our body also utilize some of the proteins as the amino acids as energy. So amino acids can be oxidized to generate like pyruvate and acetyl-CoA and enter the TC cycles for complete oxidation will produce energy, right? You guys familiar with this pathway. And also the carbons or amino acids can be utilized, can be converted to glucose and also fatty acids, which occurred under the fat condition, right? When we have a lot of nutrients taken from the diet, and we have extra amino acids, protein, then we need to convert this to the stored energy either as the glycogen or the triglyceride, right? So and TCA cycle is very important pathway that for the involved in. Try to repeat again. So TCA cycle is very important, the pathway that is involved in the conversion of amino acids to glucose or fatty acids. So here I just want to mention the gluconeogenic pathway can also be activated in the fat condition. We learned in the carbohydrate metabolism, right? You always connected gluconeogenesis to the fasting condition, right? But actually in the fat condition, gluconeogenic pathway, particularly involved in the TCA cycle, and the gluconeogenic pathway can also be activated. And the purpose of this gluconeogenesis pathway is to convert amino acids to glucose, right? And store all the fatty acids. The gluconeogenesis, it converted to glucose, right? And for the as the glycogen and stored in the body in the fat condition. All right. Any question about? Yes.
Is that more likely to happen when you have a higher protein diet, or is that always happening?
Yeah, that's a good question. So in the fat condition in particular, you may have a high protein diet, right? So your protein will again need to convert it to stored energy. So and also the other important pathway that amino acids can be used for synthesis of non-protein derivatives, for example, purine, pyrimidine, and neurotransmitters and other non-peptide hormones. Can also be synthesized from amino acids. So amino acids anabolism pathways that is, amino acids can be utilized, can be used for synthesis of proteins, and this can be balanced with the degradation of protein. So degradation and synthesis of protein always happening, and this process called protein turnover, right? And in terms of catabolism, amino acids that at the carbon metabolism that I described, can be utilized for the energy during the fasting, or can be converted to glucose or fatty acids in the fat condition. And the nitrogen metabolism of amino acids, so nitrogen can be removed from amino acids before, and carbon can be further metabolized for this purpose. So nitrogen in the amino acids can be metabolized and as the ammonia and the urea, right? And also the amino acid be utilized for the synthesis of other non-protein compounds, right? All right, so now let's talk about protein synthesis. So protein synthesis, you guys probably already know, right? When you learn biochemistry. So first of all, the genetic information need to be transcribed, right? So DNA is transcribed into 3 different types of RNAs in the nucleus. For example, messenger RNA, mRNA, a tRNA transfer RNA, and the ribosome RNA. So these three RNAs are transcribed from DNA, and it's very important, involve the translation and of the proteins. And then the second step is the translation. So once a messenger RNA is synthesized, it can be translated into a protein, and with the help of a transfer RNA and the ribosome RNA in the plasma, in the cytoplasma, right? And the three types of RNA have different functions. So messenger RNA, mRNA, usually serve as the genetic coding template, and the genetic information that in the messenger RNA determines the order of amino acids. So amino acids that protein being synthesized, and the amino acid sequence is determined by the genetic codon, right? The messenger RNA. And transfer RNA basically is the function to transfer the individual amino acids to the messenger RNA template, and for the to provide the amino acids for the synthesis of proteins. And ribosome RNA usually serve as the platform, for the translation. So there are actually two different ribosome subunits, 60S and 40S ribosome, right? You have a big and small ribosome RNA, and form the platform, right, for the translation. And this just, I don't want to go to details, but just kind of, very brief the steps. For protein synthesis can be divided into 4 phases. First of all, the amino acid activation, and then initiation, elongation, and termination. So 4 different steps. And for the amino, this is just more information about the basic components for the translation of messenger RNA. Again, we need amino acids, right, as the building blocks, and 3 types of RNAs, and ATP and GDP for providing energy for translation. And we also need initiation, elongation, and releasing factors. That is important, for the translation process, and some enzymes that catalyze the reactions in translation process, and ribosomal as a platforms. So amino acid activation. So this is a reaction for amino acids activation. So amino acid activation usually just to attach the amino acids to the transfer RNA. So then allow transfer RNA to carry the amino acids to the messenger RNA for translation, right? So and this reactions requires 2 ATPs actually. So you can see the certain amino acids attached to the transfer RNA, and then transfer RNA be able to transfer amino acids to the translation side. And the enzyme for the amino acid activation is called amino acyl tRNA synthetase, right? So we have 20 amino acids, right, in our body. So we only have the amino acyl tRNA synthetase. That means the one synthetase that catalyze the reaction for the 20 amino acids, right? We only need one, synthetase to catalyze the activation of 20 amino acids. And this enzyme's affinity for amino acids is high. That means that even when we have a lower amino acids, then this enzyme activity is high, then be able to activate the amino acids for protein synthesis, right? Okay, now post-translation modification. So once the amino acid peptide is synthesized, and more at the process is needed in order to make the protein active, right, functional. So so the process that after the translation called the post-translation modification. So a lot of different type of modification. For example, first of all, we need to remove the N-terminal methionine residues once the peptide synthesis of peptides is finished, right, complete. And then second, we need to cleave the unnecessary regions. So during the protein synthesis, some unnecessary regions also synthesized. So that unnecessary peptides need to be removed. And then this is the very important, the post-translation modification. The glycosylation means that the protein, the sugar group being added to the amino acid residues of the proteins. And glycosylation is very important modification that can regulate the protein degradation, right? So once the peptide protein is glycosylated, and this protein is relatively stable, right, resistant to the degradation. And acetylation, so acetyl group is added to the proteins. So acetylation is now, I think a lot of research now found is very important modification, can regulate protein activation. Some enzymes, in the cells that need to be acetylated, then the activity of an enzyme can be regulated by acetylation and deacetylation, right? And fatty acylation is similar, regulate the modification that also can regulate protein or enzyme activities, right? And disulfide bond formation, the protein usually contains more than one peptide, right? So multiple peptide. So peptide can be bridged by the disulfide bond. And then finally, once all this modifications finished, then protein need to be folded, right, in order again to have a function. So this folding of a protein usually occur in the ER, right? All right. So for that part, I mean, I just want to summarize. So you probably wanted to know what are the components necessary for protein synthesis. Like the three type of RNAs, amino acids, and ATP, GDP, and some enzymes and factors. But I don't think I gave you guys the specific names of enzyme, right? Just in general, these things are needed. And the fourth is protein synthesis, right? Amino acid activation, initiation, elongation, and termination. All right, so I don't think I talk about the regulation point of protein synthesis, right? But that's fine. Okay, now let's talk about protein turnover. Proteins in the cells usually constantly being synthesized or degradation. So synthesized degradation, these two processes together called protein turnover. So it is very important. The protein turnover is also not just because of some protein has a half-life, right? Some protein be getting old and need to be degraded, and new protein will be synthesized. and but also it's very important and that turnover process can also control the levels of proteins, right? So and then control the regulatory mechanism. For example, some enzyme we don't want to, and we wanted to shut down specific pathways, and so we wanted to reduce that enzyme levels. So some protein degradation pathway may be activated and to degrade that specific enzyme and to reduce the the levels of enzymes. So this is important, the regulate mechanism for the metabolic pathway, right? And the concentration of protein in cells usually determined, again, by the balance between the rate of synthesis and degradation. So, so for example, if synthesis activity is higher than the degradation, then this protein level will be high, right, in the cells and vice versa. So again, the turnover rate is very important in determining the concentration of specific proteins in the cells. And a positive balance means that can result so-called hypertrophy that increase the protein content in the cells, and negative protein balance that result in the atrophy or loss of proteins from the cells. So the negative positive balance can occur in the different stage of the life. For example, during infant, the growth stage usually will keep like positive balance, right? We need to and negative balance usually occur in the disease state, right? Like surgery or burning, kind of a disease condition, and also in the pregnant, I think they maybe also undergo like negative protein balance, right? Now, so for the synthesis, I think we just talk about very brief, the process of protein translation, right? And now let's talk about the degradation. So degradation, and there are actually three major mechanisms in the cells that can degrade proteins, and each mechanism may be responsible for the different type, the degradation of different type of proteins, right? So these three pathways includes the lysosome pathways, and proteasome pathway, and cytosolic proteinase, right? So lysosome pathway basically degrades extracellular and cell surface proteins, but also some intracellular protein. So extracellular protein, for example, some proteins in the plasma that need to be degraded. So first of all, need to be take up into the cells and then degraded by lysosome pathways. And also some cell surface protein like surface receptor. Remember we talked about LDL receptors, right? And some other receptor, of course, cell surface, and also degraded by lysosome pathways. Now let's talk about the first lysosome pathway, lysosomal proteolysis. So lysosomes usually is the organelle, right? And the membrane enclose the vesicles that formed from the Golgi complex, right? It's a type of the organelles, and inside of lysosome there are actually more than 60 digestive enzymes, right? So 60 digestive enzymes is located inside of the lysosome, and this 60 digestive enzymes the responsible for the degradation of the proteins. But here we talk about protein, but also actually lipids, can also be degraded inside of lysosomes. But here we're just focusing on the protein degradation, right? And then the substrate that can be selected by the lysosome degradation actually have 4 different pathways. Means that protein first of all need to be engulfed, to embed in the specific membrane systems. So for example, vesicle fusion. So that vesicle usually is formed through the endocytosis mechanism. For example, the LDL receptor, right, internalized and form the vesicles, and these vesicles can be fused with the lysosome, and the content inside of vesicles then be transferred into the lysosome and degraded by lysosome enzymes, right? And the second pathway is called a macroautophagy. So macroautophagy is autophagosome is another important. I think it also kind of similar to organelle, but it's a little bit different. So it's also membrane the vesicle system. So macroautophagy usually degrade the organelles, old organelle or damaged organelle, for example, mitochondria, right? So mitochondria, if you get damaged, the old, then is engulfed by the autophagosome, right? So autophagosome then fuse with the lysosome, and again the lysosome enzyme degrade the mitochondria. And the third mechanism called a microautophagy. So some proteins inside of cells can also be engulfed by the autophagosome and then fused with the lysosome. And last pathway is called a direct transporter through the chevron system. So, and what type of a protein or organelles that can be degraded by lysosome proteins? First, by lysosome pathways. So the first is I mentioned earlier that extracellular proteins, right? For example, the plasma proteins that can be taken up into the cells through the endocytosis mechanism, right? A lot of proteins are present in the plasma, right? Even hormones and growth factors and other proteins that can all be taken by the cells and through the endocytosis mechanism and degraded by lysosome pathways. And the cell surface membrane proteins that also can be used by so-called receptor-mediated endocytosis, and I give an example like LDL receptors. And then the protein that can inside of cells, right, can also be engulfed by autophagosome intracellular proteins. And lastly, the the old organelles that can be surrounded by ER membrane and form the autophagosome, right, and like older mitochondria. So again, the extracellular proteins, cell surface membranes, and old organelles, and the some intracellular protein. This are all the substrate of the lysosome protein lysis, right? So let's talk about the pathway vesicle fusion. So vesicle fusion that involve the the endocytosis of the extracellular proteins and the cell surface membrane proteins or receptors, and receptor like LDL receptors that can be degraded through the lysosomes, right, pathways. And a autophagy and pretty much just repeat, they are responsible for the degradation of organelles, right? So, and there's a whole pathway called autophagy lysosome pathway. The autophagosome can again, is very important. The vesicle system can embed the whole damaged or old organelles like mitochondria, and then form the autophagosome, and autophagosome eventually fused with the lysosome, right? And then, so the lysosomal enzyme be able to degrade the mitochondria and the proteins. And autophagy, I'm not sure if you guys heard about it, autophagy. No? Okay, so autophagy pathway in recent years, I think maybe in the past about past 10 years, it's a very hot area. The autophagy actually, the lysosome pathway is very important. The mechanism that can degrade proteins, for energy, for example, during the fasting or the starvation, the cell starvation, because of energy crisis, so autophagy activity is increased. So autophagy can degrade some proteins and to provide energy for the cell survival or something like this. Yes.
And this would just be for old or damaged organelles, it wouldn't be for organelles that are still functioning?
You mean during the fasting, during the starvation? That's a good question. So I think even not during, initially when autophagy was discovered, people think during the starvation that autophagy is activated and degrades the proteins and provide energy. But later on, actually found the autophagy is also very important even in the disease state. Autophagy even in not under fasting condition, but the autophagy lysosome activity also need to be keep at the certain levels because we always have some damaged and older proteins and need to be cleaned up. So the autophagy lysosome pathway is very important for clean the the bad or the old or damaged proteins, organelles. So if somehow the autophagy system is impaired under disease condition, that these damaged proteins cannot be degraded. So then they accumulate a lot of the dysfunctional proteins that are causing the cell stress and damage the cells and affect the cell function. So for example, in the obese conditions, then people found that autophagy activity somehow is kind of disrupted. So it cannot efficiently degrade or clean up the all kind of damage or dysfunction protein and causing the dysfunction of the cells. And this is, I think, is very good. The diagram show you the process of the autophagy lysosome pathways. So, for example, this is the vesicle. First of all, it's formed. This membrane, the autophagosome membrane, is formed from the I think it's from the Golgi system, and then the engulf or surrounded some damaged proteins, lipids, and and organelles like mitochondria, right? And then form the autophagosome. And autophagosome is formed and then fused with lysosome. And lysosome we know have a lot of 60 digest enzyme. So and then eventually forms so-called autolysosome, right? Autolysosome is formed, and the digest enzyme in the lysosome that can degrade all the content that in the autophagosome, right, the degraded protein. So this, I think, is a very nice diagram to show you the whole process, how autophagic lysosome works to degrade proteins or organelles. And I just wanted to mention that actually this year in the 2016 Nobel Prize in Physiological Medicine actually award the scientist that actually who works on the autophagy. So it's very actually in the heart area. I think it's very important. And this diagram, I think it's also important to show you, the almost like 4 or at least 2 different pathways, like vesicle fusion, 3 pathways, vesicle fusion, macroautophagy, and microautophagy pathways, right? So for vesicle fusion, basically is responsible for degradation of extracellular proteins and the cell membranes, right? So you see sometimes even bacteria depend on the type of cells. Like macrophagy, usually you can degrade bacteria, right? To kill bacteria. So and also extracellular proteins. So here just give an example of the bacteria, right? So can be endocytosis by macrophages, and then form the phagosome, and then phagosome can fuse with the lysosome, right? And so again, the enzyme in the lysosome degrade the bacterial proteins. And this is the vesicle fusion pathways, and you also there's some proteins in the plasma, in the circulation, once it get old or damaged, and then can also be internalized by the cells and through the endocytosis pathways, right, and form later endosome, and then early endosome and later endosome, and then fuse with the lysosome, right, and degrade. So, and then the macroautophagy pathways, like damaged, the mitochondria can be surrounded by the membrane structures and form the autophagosome, and this then be fused with lysosome from autolysosome, right, and degraded by lysosomal activity, enzymes. So, and the proteins being degraded, the amino acids amino acids can be used as energy, or essential fatty amino acids can be used for protein synthesis, depend on, again, the physiological state. Right. All right, any questions about this process, autophagic lysosome pathways? Okay, so and this just the additional pathway, in the lysosome mechanism, the microautophagy. So some cytosolic proteins can damage cytosolic proteins can also be degraded through the autophagy lysosome pathways, right? Okay, so do you guys want to break? Okay, yeah, we can break. Okay. Now let's move on. So I think that we finished these three mechanisms, right? Vesicle fusion, uh, microautophagy, and microautophagy. Now this is the fourth pathway. Sorry, the pathway, right? Three pathway, and then there is a fourth pathway in the lysosome degradation mechanisms. So direct transporter by the chevrons. And this pathways require the specific the amino acid sequence, the 6 amino acids right, sequence on the protein, the subject. That the sequence can be recognized by a protein, the HSC76. So HSC, he shot cognate proteins. And then also requires the specific receptors, IGP 96, and this receptor is localized on the surface of lysosome, right? And this system responsible for the degradation about 30% of cytosolic proteins, for example, glycolytic enzymes and proteinase, proteasomes, and also transcription factors. And this type of proteins can be degraded through the direct, transported via chaperones mechanism. Right? Okay, so I think that this diagram gives us a better idea, how these pathways occurs. That so first of all, again, this represents the peptide proteins, the C-terminal, N-terminals, and the pentapeptide sequence that are located at the N-terminals, and this peptide sequence is very important that can be recognized by a HSC73 proteins. And once this proteins bind this sequence on the protein, on the certain proteins, then can direct the protein to the lysosome, right, and allow the this peptide sequence bind to the receptors IGP 96. So this is represent the lysosome. So IGP 96 is a receptor on the surface of lysosome, and the HSC 73 proteins direct these specific proteins to the receptors, right? Bind receptor and allow the peptides and together with the HSC protein that internalize right in the lysosome and degraded by the lysosome. So again, this is, pathways requires the specific sequence and he shot cognate proteins 73 and also receptor, right, IBP 96 receptor, and this all things work together and be able to specific target these the proteins and degrade in the lysosomes, right? Yes.
So is that receptor required for the degradation of this stop proteins as well for the third type of lysosome?
Yes. Yes. Mm-hmm. Right. The question was whether this HSC73 protein is also degraded inside of lysosome, right? So the answer is yes. So together with the specific peptides proteins and this HSC73 protein, both are degraded inside of lysosome, right? And HSC70 protein usually present in the cytosol, right? And then be able to recognize the specific protein with these specific peptides and direct the protein to the lysosome binds receptor. All right. Okay, so here's just sort of summary of the lysosome pathways. And it's degrades extracellular proteins and cell surface proteins and also some intracellular proteins. And these are the four specific pathways that in this, that involve the lysosome pathway degradation. The vesicle fusion, basically responsible extracellular proteins, the cell surface membrane, and also, the bacteria that in specific immune cells. And then macro and autophagy, basically responsible for organelle degradation, and micro autophagy is for intracellular protein degradation and chevron-mediated pathways, and again also responsible for the cytosolic protein degradation, but requires a specific, the receptors and heat-shock cognate proteins that involved, right? And also specific peptides need to be present in that specific protein to be degraded. All right. Now the second mechanism for protein degradation is the proteolytic pathways. Is the proteasome pathways, right? Proteasome pathway that usually degrade proteins from the cytoplasma, and nuclear and ER in the cells. In general, the proteasome degradation system that degrade protein inside of cells, right? So first of all, we wanted to know the proteasome that is main the structure that degraded proteins. So and this system, proteasome system, usually degrades 80 to 90% of intracellular protein. So intracellular proteins, 80 or 90% of intracellular proteins are degraded by proteasome system, right? And proteasome, here's the structure of proteasome, contains the two parts. First of all, is the core particles. And core particles has the two sets of 14 proteins, and each alpha beta rings actually has the 7 proteins, right? You have a two set of duplicates the alpha and beta rings in the core part. And in the regulated part on each side also has 14 proteins, and 6 of them actually are the ATPase, right? So this degradation process requires a lot of energy. So 6 of them of 14 proteins in the regulatory particles are the ATPase. And also very important, the recognition of specific protein. So proteasome be able to recognize specific protein called ubiquitin. So ubiquitin is very important protein that mediates the proteasome degradation, right? So connect the protein to be degraded to the proteasome. Again, the proteasome degradation is responsible for the regulatory degradation of critical proteins inside of cells that control the cell growth, cell function like growth, proliferation, cell differentiation, apoptosis, immune and inflammatory responses, right? The cell function. So the protein that involved in the cellular functions can be degraded by proteasome, but this degradation process is regulated, but not only under the certain conditions that proteasome degradation system is activated, then can degrade this type of regulated proteins, which makes sense, right? We don't want to just depend on the cell function whether this protein need to be increased or need to be removed, or to regulate the biological process. So this proteasome activity can control cell cycles, and also can degrade, some oxidized or misfolded protein in ER, and can, regulate some concentration of the other enzymes, or the immunogenic peptides, right? And also control the transcription factors, degradation, that control the activity of transcription, right, of gene expression, and also control the cell signaling transductions. I think that this probably is a little bit easy to understand, right? Because it regulates the cellular protein that involve a variety of biological functions. So the activity of proteasome can control the cell biological process or functions. So what type of proteins, be degraded? How this protein be degraded and not the other proteins, right? How specific, this process. So first of all, the proteins that can be degraded usually have a specific amino acid sequence, right, motif. And some altered structures like oxidative modification or exposure of the hydrophobic amino acids. So this kind of altered structures or alter the proteins that be able to recognize by proteasomes and degraded by proteasome. And also this actually is very important pathway that's called ubiquitin. It's a major pathway that regulate the protein degradation by proteasome. So that means the ubiquitin need to attach specifically to certain proteins, and only that protein has the ubiquitin attached that can be recognized by proteasome and be able to degrade it by proteasome, right? So this again is a major mechanism that for the proteasome degradation, right? So proteasome only degrade the protein with ubiquitin attached, not the other proteins, right? So again, this is the major pathway mechanism. So now let's talk about ubiquitin. So ubiquitin is a small protein. It's a high conserved and present in almost any type of cells, right? So any type of cells have a ubiquitin. And ubiquitin present in the cytosol of the cells. And the function of ubiquitin is to target proteins for degradation. Like I said, ubiquitin has to attach to certain damaged or whatever dysfunction proteins, and then allow proteasome be able to recognize them and degrade them, right? So usually, the protein that unassembled, the cell always will make mistake, right? Some proteins are synthesized but not, unassembled, and this type of protein need to be degraded, and also damaged and misfolded proteins, and short life, the protein with short half-lives, right? So this type of proteins be able to, ubiquitin be able to recognize, attach to this type of protein, and for the degradation by proteasome. Questions? No, just suggestion. All right. So, and the regulation of proteasome activities. Um, so phosphorylation of certain domains in the target proteins. So, for example, that proteins somehow, in certain range or certain amino acids get phosphorylated, and this phosphorylated proteins uh, can stimulate degradation. So proteins get phosphorylated, the more easy to be degraded, right? And glycosylation, I just mentioned earlier, this inhibits degradation. So once the protein gets glycosylated, and it kind of resistant to the degradation, right, to protect from the degradation. So degradation can be regulated by phosphorylation and glycosylation. So phosphorylation promotes degradation, and glycosylation inhibits degradation, right? And the degradation of protein by proteasome can also be regulated by hormones. So insulin basically inhibits proteasome activity, so inhibits proteasome degradation, and glucagon, corticoid, and stimulate degradation. So when, for example, under the fasting conditions or some kind of stress condition, that the the degradation is proteins promoted, right? Which makes sense. We probably wanted to break down some proteins and for energy, right, in the fasting, and also some disease injury usually also activate a protein degradation. For example, the burning or surgery, right? So you promote the protein degradation, so will activate the proteasome activities. Right? Any questions? Okay, so the last mechanism for protein degradation is the cytosolic proteasome proteinase, right? So inside of cells, there are also a large number of the different type of proteinase that can also directly degrade proteins. And here, I just want to mention that calpines, right, a set of proteins, proteinase. Calpines activities is depend on the calcium, right? So calpin can degrade usually the cytosolic proteins, and in some conditions the calpin is not active, right? We don't want it to always like degrade protein. So calpin is not active. So the two things can regulate calpin activities. One is the calcium. So calcium actually activated the calpin's activities. And the other thing is the factor is is a protein, the inhibitory protein called calpacidin. Calpacidin is the inhibitors of calpins. So one, and let me just go to the next slide. It will be easier to understand, I guess. So so calpin, so this represents the calpin molecules, right? And in some conditions, calpin is bind to the calpacidin, right? So once it's bind to the calpacidin, the calpin activity is inhibited, is not active. And once in some conditions, when cell calcium concentrations increase, then can stimulate this association of calpin with the calpacidin. So calpin is released and activated and be able to degrade some proteins inside of cells, right? So calpin, again, is the type of proteinase in the cells that the activity can be regulated by calcium and inhibitors, calcistatin. All right, I think that's pretty much all that I have for the protein synthesis and degradation. And just to summarize, and for the protein synthesis, we just talk about the protein translation from how protein synthesized, right, from the DNA to transcribe to three major RNAs, messenger RNA, transport RNA, and ribosomal RNA, and each type of RNA has the different function. Messenger RNA basically serve as the coding template and determine the orders of amino acids in the peptide and proteins. And then we also talk about the process of translation. Amino acid activation, initiation, elongation, and termination. And I think we focus on more the protein degradation, right? So protein degradation, protein can be degraded through the three mechanisms. And first is the lysosome pathway, and second proteasome pathway, and then third is the proteinase, right? The activity. So in the lysosome pathways, and there are four mechanisms of pathways called vesicle fusion, which is responsible for the degradation of extracellular and the cell surface proteins, right? And the second is the macroautophagy, responsible for the degradation of old organelles, and we have an example like mitochondria. And the third is the microautophagy, and usually degrades the intracellular proteins. And the last is the directly transport, right? By chaperones that involve the specific peptides that can be recognized by C73 proteins, and then these proteins direct the target protein to the lysosome and bind to receptor, the the receptors, right, on the lysosome, and then degraded by lysosome. And inside of lysosome, there are actually 60 digestive enzymes, right, can respond for the degradation protein. And the second pathway is the proteasome degradation pathway. Proteasome, we have a specific structure, right? You have a core and then the regular particles. And the proteasome degrade proteins, the specific degrade protein through the ubiquitin mechanism. So ubiquitin is a protein that can bind the specific protein that to be degraded, and ubiquitin can be recognized by the proteasome, right? So, and be degraded. And we just talk about the proteinase, the calpain, right? And calpain just directly regulates intracellular proteins, and its activity is regulated by calcium and the inhibitory proteins. That's, I think, all for the protein synthesis and degradation. I think that Carrie is going to talk more specific about the amino acids metabolisms and how the carbon of amino acids metabolize and nitrogen metabolize the urine cycle and stuff, right? Let's see what else. Today is the due date for the peer review form, right? And I know some of you have a question whether you need to turn in individually or a group. I think in one group I just need to turn in one, right? Review form. Any other questions that you guys have? Yes.
When are the exam dates going up?
Oh, okay. Yeah, I'll talk to Tia and ask her to post it. I think it should be pretty soon this afternoon, I hope. Yeah. And what else? Yeah, and we have about a half hour, so you guys can work on peer review form or something like this, right?